Um, Today, it's a pleasure to have Soojin from biology give our breakfast club talk. Everyone may know Soojin. She came here in 2004. Her first two degrees were at Seoul National University in Korea, and then a PhD from the University of Chicago. I didn't realize until I took a look at her CV that she did a little bit of middle school biology teaching back in Korea.
I did.
And she also spent some time, it's unclear from her CV how much, in Scotland. Apparently it wasn't enough to give her a Scottish accent, but...
I could try if I wanted to, but
you could
you
try?
probably don't want to hear it.
How long were you actually there, Soojin?
About a year.
About a year.
Yeah.
Okay. Anyhow, she came in 2004. We're proud to have her as a colleague in our IBB faculty. And Soojin, it's all yours.
Thank you. Thank you. Thank you, Bob, for this opportunity, really. It's really nice. And James and Coley and everybody for organizing this and for wonderful breakfast. It's really nice. Okay. Okay, so I'm going to talk about one of my...
Can I use the microphone just out in the back because it's so loud?
Oh.
Is it on?
It's on for him, I think.
It only records for me.
Right.
Ah, okay. I thought you had the...
I could try to yell.
Can you hear her in the back?
Can you guys hear me in the back? I tried to yell. Okay. Okay, so I'm excited to talk about one of my main research foci in my lab about DNA methylation and genome evolution. So many of us in the room who may not be in evolutionary biology probably heard about DNA methylation because it is one of the most important types of epigenetic modification. Epigenetic modification means chemically marking DNA or chromatins and so on so that the cell can regulate in addition to the information already in the genetic codes. And DNA methylation is really widespread in all the three domains of life. It's found in bacteria, eukarya, and also in archaea, although it's not clear if the archaea one is originally there or was laterally transferred. I'm going to talk about one type of DNA methylation, which is cytosine DNA methylation. Okay, as we all know, there's 4 bases, and one of them, cytosine, looks like this. And the enzymes that put methyl groups, DNA methyltransferase, puts the methyl group to the 5 position. So instead of H, now you have CH3, and this is the template. Okay, and this only occurs for C's right next to G in the 3' direction. So these nucleotides are called CPG. So only C and CPG are methylated. And this is the predominant mode of DNA methylation in animal genomes. In plants, there's maybe some other ways. For example, they use C and G. So N means any of the 4 bases. But in animal genomes, it's almost exclusively a CPG, and this makes analysis for us easier than otherwise. So there are many studies going on about the role of DNA methylation, and the emerging theme from these studies is that DNA methylation acts as a silencer or dimmer of gene expression. Okay, the actual mechanism how it does it is still debated, and we'll have a couple of slides describing some mechanisms. But basically, DNA methylation can silence gene expression, and it can also play a role in chromatin compaction. And it can suppress recombination between transposable elements or repetitive sequences. And because of these things, these are related to genome stability. Okay. Also, DNA methylation is the main mechanism how one of the X chromosomes is silenced in females. Also, it plays a role in genomic imprinting. By putting methyl group in C, you can distinguish now between C versus methyl C. So not only you have 4 bases, but you have additional information. And you can distinguish this way 2 alleles from father and mother, and that's how the genomic imprinting works. So these are all functional aspect of DNA methylation that has been widely studied in mammalian models, human and mouse, basically. But another important aspect of DNA methylation, which is not always appreciated by molecular biologists, is the fact that it causes mutations and is actually a main source of mutations in animal genomes. And if you cause mutations, it leaves evolutionary signature. So it is a very important source of genome evolution as well. And I'm going to show you what I mean by that. First, let's see how methylation causes mutations. So this is the methylation that I've shown already. So cytosine gets methylated, becomes methylcytosine. And this molecule is chemically unstable. Okay? So it loses the amine group in here very quickly by spontaneous chemical reaction. No enzyme is necessary in this step. So this step occurs very quickly. And then when you lose thymine here and you insert the water molecule so you become oxygen instead of amine, then chemically it's equivalent to thymine. So what they have done is that the cytosine became thymine by methylation and deamination. Okay? So C to T mutation has occurred. And this occurs very frequently. Now, DNA repair mechanism should take care of this, but it's not very efficient because T is naturally found in the genome and it doesn't always distinguish between the T that is mismatched versus T that it should be the real T. So this is, even though DNA repair exists for this mechanism, because this is so rapidly occurring, there's still a lot of mutations that goes through to the next generation that generate from this process. So basically C to T mutation at CPG sites will create a CPG to TPG, or in the reverse trend it will be CPG to CPA. So these type mutations are called as CPG mutations. Okay? And these are really the major source of mutations in animal genomes. I've done some work in my lab to see how often these mutations occur compared to other C to T or G to A mutations. And we have shown that it's about 15 times higher than the normal transitions. And in other animal taxa, there's a wide variety of estimates, but everybody estimates at least an order of magnitude greater than the normal mutations. And in the chimpanzee genome sequencing paper, it was shown that 1/4 of all point mutations between human and chimpanzee has occurred at CPG sites. So you can appreciate how important these mutations are in animal genomes, including our own. Now, one of consequences of this process is that many CPGs will change to TPG. In other words, the CPGs will be eliminated from the genome. So if you look at, for example, human genome, we don't see a lot of CPGs actually because they're all changed to TPG or CPA at some point and then went through other mutational processes. So DNA methylation will, in effect, reduce the frequency of CPG dinucleotides. And this is an important property that we'll be using in next steps. So in my lab, we have been analyzing the influence of genomic methylation on evolution for a few years now, and we have several directions we have pursued. We have shown that methylation influences genomic molecular clock and also affects compositional evolution. Now, recently we have started looking into the functional aspects of DNA methylation and influence on genome evolution. So these are the two papers that we have submitted and focusing these days, and I'm going to talk about these two today. So let's first talk about methylation and evolution of vertebrate promoters. Now, for the rest of the talk, I'm going to introduce one measure that you need to know, okay, and that is normalized CPG contents, or I say CPG OE for simplicity. CPG OE is a simple measure that basically is the ratio of observed CPG in the data normalized by the expected CPG based upon the random association of C's and G's. So if you have a lot of methylation, then the CPGs are changed to TPG or CPA, so this value will decrease, and this CPG OE will decrease. So if you have a lot of genomic methylation, CPG OE will decrease. And if a